Right. I wanted to first start off by saying thank you for having me. I want to start off with a self-introduction because this is my first presentation and hopefully not my last presentation. My name is Martin White. I'm a second-year master's student at the University of Tokyo. The first year was dedicated to making sure all of the necessary measurements were taken, and the second year will be dedicated to the execution of the experiment. So let's begin. So this will be the general layout of the proposal that we're going to be going over today. So first, we're going to start with a brief introduction of magnetoreception. So through extensive research, it was noted that certain animals have the ability to sense the Earth's magnetic field and extract compass information. For the purpose of this proposal, however, we're going to be focusing on the migratory bird because their magnetic compass has been well studied and well documented. For the migratory bird, there are a few key features which are important for this research. Firstly, that depending on the wavelengths of light, they're still able to maintain their magnetic sensing ability. It was demonstrated that in blue and green light, it's able to maintain this ability. However, in red light, it is not able to do so. Secondly, it is able to detect changes in the magnetic field. And last but not least, it is unable to discern the polarity of the geomagnetic field. However, it is able to discern the axis of the field, thereby making it an inclination-based compass. So we're going to move on. So we're going to move on to RPM and possible shortcomings. As for the most part, the radical power mechanism has already been explained by Professor Orr. There's just one key takeaway that I would like to go over. Photochemical generated radical pairs are magnetically sensitive and can be used in magnetoreception. And that's one of the things we're going to focus on in this proposal. So, we wanted to understand how this works anywhere. So in 2000, Shelton proposed that there's a specific protein in which is used as a magnetoreceptor, later named cryptochrome. As we know now, cryptochrome is a flavoprotein, which is involved in circadian rhythm regulation, and also, after exposure to blue light, it forms radical pairs within inside the molecule. And we can demonstrate that right here. So you see, after the cofactor flavin-adenine dinucleotide is excited by the blue light, it goes into a photoexcited state. Then after, the first tryptophan donates an electron, and then you have a subsequent electron hopping from the rest of the tryptophans, thereby forming a singlet radical pair. So now, we wanted to know how this works. So they already performed research on this topic in general. From this research, all observed field effects occurred at magnetic fields higher than that of the geomagnetic field. Vertebrate cryptochrome MFEs were observed to be weaker than that of their plant counterparts, and that was rationalized as with the vertebrate cryptochromes, they have an extra tryptophan, and that is rationalized versus the tetrad versus the triad relationship. Last but not least, they're unable to observe MFEs at room temperature. So, with these facts in mind, there are a couple of questions that do come to mind. Firstly, is it a different molecule that might be at play? Is it a different mechanism? Or the question in which we believe, is it the environment in which is playing a key role here? As is the nature with most biochemical reactions, the environmental pressures can change how the reaction takes place and even the concentration of those reactions. So, with that in mind, if we can say this a lot better, are there differences in cryptochrome photochemistry and magnetic sensitivity between the in vitro reactions from the previous research and the in vivo experiments that we would like to perform? Secondly, will these in vivo measurements add more information to support that the avian magnetoreceptor is using this mechanism, or the opposite, of course? And lastly, where is cryptochrome localized within the cell? As previously mentioned, due to different environmental pressures, reactions can change. So depending on where it is localized within inside the cell, then it stands to reason that the reactions themselves might also change. So in terms of reactions and in terms of concentration, there are a lot of different changes that we might be able to see depending on where it is inside the cell. So we're going to move on to the research proposal. So the reason I'm here today is because we want to answer those exact questions with this following technique. We're going to be utilizing these two forms of microscopy. First, fluorescence microscopy, and secondly, transient optical absorption detection and/or code. First, for fluorescent microscopy, we're going to be using it for cellular observation of the cryptochrome expression localization. While, as remarked in previous presentations, while the reactions themselves might be too quick for might be too quick to notice via fluorescence. However, we can still use it to see where they are inside the cell. There still would be enough fluorescence for that as well. Also, while we are making these measurements, we then have the possibility, more specifically, to make MFE measurements. While we don't know whether or not we will be able to, the possibility still does exist. So next we're going to move on to transient optical absorption detection. So as mentioned previously, we now answer the first we now answered one of the questions of the localization. Now we want to know what's happening inside the cell via the photochemical reactions. With the utilization of TOAD, we have direct photochemical observations of the reactions via their intermediate radical absorptions. Secondly, we are able to use flash photolysis. As these reactions are quite quick, when we're utilizing flash photolysis, we are able to cycle these reactions so we can perform it quite quickly. So now, before we get to this part, we then need to prepare the samples. So, with this proposal, we're trying to make sure that everything is done as simple as possible. So we take ErCry4 plasmid, mix it with a transsection reagent, shake it a little, pour it on top of the HeLa cells, and it will transiently transfect the culture, usually within about 24 hours via liquefaction. The reason why we chose HeLa cells is because as a model organism, they are quite well studied, and at the same time, as cancer cells, they are quite robust and resilient. As while we are introducing new environments and even new changes into the experiment, they will then be able to take the stress of anything that we might throw their way. So now, we want to answer the question, why are we studying European Robin Cryptochrome 4 or ErCry4 and not ErCry1 or ErCry2? As mentioned previously, we want to focus our proposal on migratory birds, such that if we are focusing on migratory birds, we would want something that's useful during the migratory period. And as it was shown, ErCry4 is upregulated and/or the expression is increased during the migratory period. Secondly, it is localized within the robin's retina. As we mentioned previously, it requires blue light to be activated, such that the best way for it to receive that blue light then would to be in the eyes where we normally receive the visual information from sunlight. Thirdly, the genetic expression remains unchanged during the circadian rhythm cycles. So why is this important? Well, we mentioned before that we could use ErCry1, ErCry2, or ErCry4. In this case in particular, ErCry1, ErCry2 shows an upregulation during the circadian rhythm cycles. However, ErCry4 does not. So of the few known utilizations of cryptochrome, we know that it's used within triggering the development stages of plants and animals. We do know, as I mentioned previously, within the circadian rhythm cycles, and also as we're proposing today, as a magnetoreceptor. And as you remove the first, that it's not being used inside the circadian rhythm cycle, you then go on to you can make the assumption that it's used for something else or it plays a different role. So, last but not least, the important structures are maintained. What does this mean? This means nothing until I go to the next slide. So in 2016, Kuta proposed that in order for a molecule to be able to bind FAD, it would have to have the required 4 amino acids in the right places. So you see here, if you look at E, you'll see that this is a comparison between ErCry. ErCry has the 4 amino acids in the right place, and if you compare that to the European Cry4, it only has 3 of those amino acids in the same places as ErCry. However, the remaining amino acid, as you can see here, R298, is closer towards the FAD binding site. What this means is that
you can use the other
okay. So it's closer to the FAD binding site. So what this means, it has a much stronger FAD binding potential, and it's something that we're looking for this research. So to sum up, we have the transiently transfected culture, and then we can use fluorescent microscopy for cryptochrome localization and MFE measurements. And then we have the toad microscopy, which we can use for intermediate radical detection and flash photolysis. So, in summary, the observations on MFEs in vitro may not be telling the whole story. So we want to be able to see if a changing environment will happen. So we propose studying the MFEs of cryptochrome within the view to help address this discrepancy. Thirdly, we want to use liposome transfection techniques, which will be used to introduce cryptochromes into HeLa cells to undertake such studies. Last but not least, cryptochrome photochemistry and MFEs thereon will be studied in a time-resolved manner using our unique toad microscope approach, which observes intermediate radicals directly. Sorry, I didn't intro you. So just in terms of acknowledgements, I just want to say a special thank you to my group members. Without them, this work would not be possible. They have saved my life multiple times. 2 of them, which are not here right now, Sahili, she told me to say, Gaius is better than Arthur. I don't know what that means. She told me to say it word for word. And Norbert, who has helped me more times than I can ever count. I would like to thank the next One Book Show Scholarship for allowing me to work. And lastly, thank you all for listening. And if you have any questions, let me know.
questions and comments.
I just wasn't quite sure why it's interesting to know the localization of ErCry4
Mm.
in cells which have nothing to do with birds?
Well, as you mentioned before, it may not have nothing. It may not have anything to do with birds. But the more information that we have on the reactions in different environments will give us more information on how it works. So even if it's in a different cell, and as we plan to do later, to introduce in different cells and probably in different tissues if possible, then we will be able to just have a much better background understanding of how they work and how the reactions change. So it just makes it easier to then assume how it will react with inside the retina. So we're just kind of building, for lack of better words.
And will you be able to compare it with the purified protein?
Yes, that's with a lot of the results previously made. They were made under a time-resolved system, as which we are also using the same. So as we are making these new, as we're receiving this new information, we're then able to have that direct comparison because we're using not relatively the same, but the time-resolved measurements.
Thank you.
Can I just comment on that? So one of the nice things about using Toad will be that we can, in principle, compare directly with the in vitro measurements because we're using the same technique. Right now we can't. I mean, in the preliminary sense, we are limited to single absorption wavelengths. So we use a probe laser. So we can probably select 3 or 4 wavelengths by using different diode lasers at the moment. We don't have the direct comparison that you have from the various experiments you have in Oxford to give you the full wavelength dependence. Okay, but in principle, if we are hoping in the future that we will be able to install a supercontinuum laser in this microscope so that we can have the full wavelength dependence. But right now we can't do this. So this is really just preliminary measurement in that sense.
So, time's over. Let's thanks all the speakers. I'm sorry I didn't keep time, and next session from 25. Half past 4.